All genetics research requires DNA. DNA stands for deoxyribonucleic acid. It's the blueprint of life. But where is the DNA and how do you get it? Our genomic DNA exists inside the nucleus of our cells. There are also pieces of DNA inside some of our organelles, such as the mitochondrion and the chloroplasts in plants. If you take a look at our genomic DNA, it is nicely organized into chromosomes inside the nucleus. Humans have 23 pairs of chromosomes, but not all animals have the same number of chromosomes. If you look at the hermit crab genome, it has 127 pairs of chromosomes. If you look at a species of ant, the Australian ant, it has 1 pair of chromosomes. The type of genome a scientist wants to look at, or the gene they're interested in, really depends on the questions they're trying to answer. So let's take a closer look at an animal cell. The nucleus is in the center of the cell, and the nucleus is actually surrounded by a nuclear membrane. If you look at the outside of the cell also has a plasma membrane, and that's made up of a lipid bilayer. Inside the cellular membrane is where we find the cytoplasm. This is where all the action happens. We've got lots of enzymes zipping around. We've got lots of organelles, such as the mitochondrion. We've got the rough endoplasmic reticulum, the smooth endoplasmic reticulum, lots of vesicles. There is a lot happening inside the cell. It's a very, very, very busy microenvironment. If you look closely at that membrane on the outside, it has lots of little pores in it. And this allows chemical molecules to move in and out, and these are important for communication, growth, and cell division. We are able to extract DNA from cells using a special combination of buffers and enzymes. Breaking open a cell with detergent is a bit like doing the dishes. We use hot, soapy water because that detergent is really good at breaking up fats. The detergent breaks open the cell and the contents spill out into the tube. There are lots of different kits you can buy to do DNA extractions, but all kits basically do the same 4 things. Firstly, you break open your cells with a detergent. Secondly, you have to get rid of all of that protein, so we digest it or break up all the protein with proteinase K. Thirdly, we do some wash steps to get rid of all of the protein and the cellular debris that can actually inhibit your downstream reactions. And then finally, we dissolve our DNA, and that DNA is nice and pure and it's ready to go in your reactions.